Ever ask yourself, what's the secret behind life's instruction manual? Nucleic acids featuring DNA and RNA hold the key, guiding the growth and functioning every living thing. Want to know how these tiny molecules shape the incredible story of life? My name is Kirei.
My name is Enzo.
And I'm Leonard Shape.
I'm Lela.
I'm Tiffany. I am Rana.
And we will be discussing the world of nucleic acids.
Friedrich Michels discovered nucleic acids through his investigation into the chemical composition of cells. In 1869, he isolated a substance from the nuclei of white blood cells obtained from passhook bandages at a local hospital. This substance, which he called nuclein, was later recognized as nucleic acid. Michels' work involved meticulous chemical analysis where he identified a new class of compounds or nucleic acids within the cell nucleus. His groundbreaking discovery laid the foundation for the role of nucleic acids as a carrier of genetic information. Although their true significance took time to be fully comprehended by the scientific community. So what are nucleic acids? They are biopolymers or small biomolecules essential to all known forms of life. They are composed of monomers, which are nucleotides made of three components: a phosphate group, a 5-carbon sugar, and a nitrogenous base. If the sugar is a simple ribose, then the polymer is RNA. If the sugar is a ribose derived from deoxyribose, then the polymer is DNA. They function to create and encode and then store information in the nucleus of every living cell of every life form organism on Earth. In turn, they function to transmit and express that information inside and outside the cell nucleus, to the interior operations of the cell, and ultimately to the next generation of each living organism. The encoded information is contained and conveyed via the nucleic acid sequence, which provides the ladder step ordering of nucleotides within the molecules of RNA and DNA. Nucleic acids serves as the foundation of genetics, playing a vital role in inheritance, evolution, and the functioning of living organisms. They dictate the synthesis of protein and regulation of various cellular processes, shaping the characteristics of all life forms. Beyond their role in genetics, nucleic acids have diverse applications in biotechnology, medicine, and research. Techniques like PCR or polymerase chain reaction and genetic engineering rely on manipulating DNA and RNA. They are also crucial in developing diagnostic tools, gene therapies, and understanding disease mechanisms. Now let's go deeper in understanding the science behind nucleic acids by explaining the main complex classes of them, the deoxyribonucleic and the ribonucleic acid.
As said earlier, DNA functions to create, encode, and then store information inside the nucleus of every living cell. It carries genetic information essential for the functioning and development of a living organism. It can be found in all eukaryotic and prokaryotic cells as well as in many viruses. This just means that all living organisms have DNA. The DNA's main job is to store genetic information in the form of a code. And this information is crucial for the transmission of inherited traits from one generation to the next. DNA is passed down from one generation to the next during reproduction, ensuring the transmission of genetic information. Offspring inherit a combination of genetic material from their parents, leading to the continuity of traits within a species. The genetic information in DNA can influence an individual's physical characteristics, susceptibility to diseases, and other aspects of their biology. The genetic code in DNA serves as the basis for the synthesis of proteins, which are these molecular machines and structural components essential for the functioning of a living organism. DNA acts as a blueprint or a recipe for a living thing. Though you might be wondering how can something as small as a molecule be a blueprint for something as complex as an elephant, a tree, or even bacteria? The answer lies in the structure of DNA. DNA is the molecule that carries genetic information essential for the functioning and development of an organism. DNA is made up of two linked strands that wind around each other resembling a twisted pattern, a shape known as a double helix. Each strand has a backbone made up of sugar, deoxyribose, and phosphate groups. Attached to each sugar is one of four bases: adenine, cytosine, guanine, and thymine. These bases are paired together in the middle and are held together by hydrogen bonds. The sequence of the bases along the DNA's backbone encode biological information such as the instructions for making a protein or RNA molecule. It also determines the sequence of amino acids in protein through the genetic code. This unique sequence of amino acids result in the production of specific proteins that contribute to an individual's physical and biochemical traits. Now moving on to DNA-based pairing.
Now, let's buckle up and unravel the intricate process of DNA-based pairing. But first, to define what DNA-based pairing is, we need to understand what a base pair is. In a DNA, a base pair is a link between nitrogenous bases that aids in helping to hold together the two nucleotides. Now, complementary base pairing is referred to as the match-up of specific nitrogenous bases in the genetic material. There are certain rules that must be followed when connecting the two strands of genetic material into the double helix configuration of DNA. The double helix typically looks like a ladder twisted, while the bases act as the rungs of the ladder. The DNA molecules contain the nitrogenous bases adenine, guanine, cytosine, and thymine. Chargaff's rules pattern explains how these bases fit together. It explains why adenine always pairs with thymine and cytosine pairs with guanine. It is named after the scientist Edwin Chargaff who discovered this pattern. He found that the proportion of adenine to thymine and cytosine to guanine were generally the same in all DNA molecules. He developed the base pairing rules wherein he stated that when one complementary base was present, the other side of the double helix had to contain the other. Since adenine and guanine are composed of two nitrogenous rings, it is considered purine. While on the other hand, cytosine and thymine is composed of one, so it is considered pyrimidine. Pyrimidines and purines always form a pair in DNA in order to maintain the same molecule species. So, how do these bases bond together? The nitrogen atoms, which are referred to as nitrogenous bases, allow them to pair up.
Because each nitrogen has a pair of electrons that make it slightly negative, it can be attracted to the slightly positive hydrogen atoms on its partner.
This is called hydrogen bond. There is a specific place in the sugar phosphate backbone of the DNA where each base pair binds.
The two vertical sections of the ladder represent the two backbones. Between the base on one strand and its pair on the other strand, hydrogen bonds are formed.
Cytosine and guanine are formed with 3, while adenine and thymine pairings are formed with 2 hydrogen bonds. In addition, hydrogen bonds are weak. They can be easily broken when it's time to copy DNA. Enzymes break these bonds, unzipping the DNA's double helix into 3 separate strands.
Because each base pairs with a specific partner, the enzymes that assemble the new strand or the daughter can read the old strand or the parent and use it as a guide.
To sum it all up, we tackled that when nitrogenous bases form with hydrogen bond with one another, a base pair are created. Because of this, base pair has a designated partner. Now, hydrogen bonds are weak, allowing the DNA to unzip, and this lets enzyme replicate the DNA. Now, Primary Santos is here to unveil the important or crucial role RNA plays.
Ever wonder why DNA gets all the attention? It's like a popular kid with its double helix stand, a twisted ladder holding all the secrets of your traits. But guess what? RNA is the real hero behind the scenes. Without RNA, the whole genetic message would be lost. So let's appreciate RNA, the unsung hero, just as crucial as the famous DNA. RNA or ribonucleic acid, from the word itself ribose, it is a type of sugar, and nucleic acid a type of biomolecule. Diving deeper to its definition, RNA or ribonucleic acid are transcripts and regulators of expressed genetic information.
RNA or ribonucleic acid refers to molecules produced during transcription, acting as carriers of genetic information that help make proteins in our cells. Additionally, this serves as regulators of genetic information that controls the amount of proteins produced in our bodies. Another definition for RNA is that these are usually single-stranded, made of ribonucleotides that are linked together by phosphodiester bonds. A quick recap. We all know that nucleic acids have monomers, and the monomers for nucleic acids is called nucleotides. Both DNA and RNA have nucleotides which consist of three parts: the sugar, phosphate, and the nitrogenous base. RNA is usually single-stranded and it is comprised of ribonucleotides linked together by phosphodiester bonds. A ribonucleotide in the RNA chain contains of ribose, one of the four nitrogenous bases, and a phosphate group. Now, let's move on to the four major types of RNA. In eukaryotic cell, DNA generally stays in the nucleus, but the messenger RNA has the ability to leave the nucleus, carrying the genetic message to the ribosomes. Ribosomes make proteins, and RNA is actually a major component of ribosomes. This is called the rRNA, which stands for ribosomal RNA. Next, we have the transfer RNA or the abbreviated tRNA. This is tasked with transferring amino acids to the correct mRNA codon. And finally, we have the microRNA, being the smallest type. These regulate gene expression by interfering through mRNA messages. In conclusion, RNA plays a vital role in carrying genetic information, ensuring proper alignment, delivering correct amino acids, and regulating gene expression through interference of specific mRNA messages. To deepen our understanding of the intricate world of RNA, we have Ms. Tiffany Santiago to discuss the base pairing of RNA.
Now let's take a fascinating journey into the world of RNA and explore the dance of molecules within its structure. At the core of RNA structure are its four bases: adenine, uracil, guanine, and cytosine. Now imagine them as characters in a dance, each having their own unique partners. Adenine and uracil waltz together while guanine and cytosine pair up for a tango. Quite silly, but these are also what we refer to as complementary bases. These pairings are then maintained by hydrogen bonds. Subtle yet crucial forces. Hydrogen bonds act as molecular glue binding the complementary bases and stabilizing the overall structure of RNA. Adenine and uracil are connected together through two hydrogen bonds, while guanine and cytosine are linked together by three hydrogen bonds. Now, within the structure, the major and minor groups emerge as the cavities between the paired bases. Major being the larger and more spacious, minor being the smaller and more narrow. Now, these groups aren't merely just spatial gaps. They introduce opportunities for molecular recognition. The asymmetry of these groups influence how proteins and enzymes engage with the RNA molecule. Which leads us to this question: who are the spectators of this dance? The answer to that? Proteins. Now, they come in recognizing the unique moves of major and minor groups. These molecular actors recognize and interact with the specific sequences within the structure of the groups. These interactions are actually pivotal in processes such as transcription and RNA modification. The coordinated interaction between the genetic material RNA and proteins are actually crucial in making sure that the cell runs smoothly. But did you know that the angle between the adjacent base pairs can actually vary? There isn't a standard angle. Well, this is because the structural characteristics of RNA is actually more diverse than we think. This is influenced by its single-strand nature and complex folding patterns. Additionally, although RNA is known to exist as a single-strand molecule, it can actually briefly fold back into itself and create short-lived double-stranded structures. Now, in concluding this exploration, we have unraveled the subtleties of RNA base pairing and the architectural significance of major and minor groups. These molecular intricacies form the foundation of genetic information transfer and cellular regulation. Now, let's have Ronas and Pedro for the final look into the features of DNA and RNA.
Now, let's delve in the realms of the function and structure of DNA and RNA. Deoxyribonucleic acid, or DNA in short, is a molecule that carries genetic information for the functioning and development of an organism. It is located in the nucleus of a cell. DNA is made of two linked strands that winds around each other that resembles a twisted ladder. It is in the shape of a double helix. Each strand has a backbone made of alternating sugar called deoxyribose and phosphate groups. Attached to its sugar is one of the bases, adenine, thymine, cytosine, and guanine. The two strands are connected through chemical bonds between the bases. Adenine bonds with thymine, cytosine bonds with guanine. The sequence of the bases along the DNA backbone encodes biological information such as the instruction for making RNA molecules or proteins. For ribonucleic acid, or RNA in short, it is a vital molecule involved in protein synthesis. It plays a crucial role in cellular processes by facilitating the creation of proteins through translation. It is mainly found in the cytoplasm. RNA is similar to DNA in structure, but they have different cellular function. Similar to DNA, RNA has the same basic elements, such as the sugar phosphate backbone and the four bases. However, there are three major differences that take account for the different function between the two molecules. Firstly, RNA is a single-strand nucleic acid, which is more unstable than DNA, but it allows flexibility to bend back on itself to form weak bonds to another part of the same molecule. Secondly, RNA has the sugar ribose instead of deoxyribose in DNA, which is important for enzymes that distinguish DNA from RNA. Lastly, RNA has the uracil nitrogen base instead of thymine, which allows to form the base pair complementary rule. Adenine binds to uracil and guanine binds to cytosine. To sum it up, DNA is a genetic blueprint in the cell's nucleus. It is in the shape of a double helix and it has specific base pair, while RNA is located in the cytoplasm and it is a single-strand nucleic acid, has ribose sugar and uracil. These molecular differences give them different jobs in the cell. Together, they play a crucial role in the cell activities.